We do have the pleasure of having Dr. Gary Samuelson on this call tonight. Pleasure to stand beside him on this journey. I personally, physically wouldn't be here without this breakthrough technology. Dr. Gary Samuelson is one of only a handful of atomic medical physicists in the world who led a team of scientists and managed to stabilize these redox signaling molecules outside of the body, stabilized, balanced exactly the same way they're found in healthy cells inside our bodies. So it's an absolute pleasure for us to have Dr. Gary Samuelson himself on this call tonight. I'm going to hand over him to do a short presentation, and we can open up for a Q&A once he finishes. We do have a chat box if you want to ask questions for the chat box as well. So welcome, Dr. Gary, and thanks for coming on tonight.
Thank you very much. It's a pleasure for me to be here. It's also nice that you take some time out of your evenings to learn a little bit about science, because you'll find probably that's all that will be able to speak to you about this evening. If it's all right, we can start with a 10-minute presentation on a review of redox signaling technology as it stands right now in the world, if that's fine. Well, I'm Dr. Gary Samuelson. I have a PhD in atomic medical physics, and it's a cross-disciplinary program also with the medical department, and so that I spent my career advising and investigating different medical technologies in the field. One of the most interesting medical technologies, and probably the most fruitful one, is a technology called redox signaling. Now, I'll explain it a little bit. It all starts with an oxygen molecule. As an atomic physicist, this is the way we look at an oxygen molecule. It's a nucleus. It has some protons inside and then electrons that are on the outside. Two of these electrons are unpaired electrons. They're called free radicals, and they cause the oxygen molecule to have special properties. One of the properties of an oxygen molecule is that it can be oxidized. And we're going to be talking about different oxidation states of oxygen tonight. Oxygen itself is in an oxidation state that if gaining electron would put in a different one, it would become a superoxide anion. As it gains another electron or is reduced, it becomes a peroxide anion, and then a hydroxyl free radical and also a hydroxyl ion and other such species are altogether known as what they call reactive oxygen species. They're species that have the different oxidation states of oxygen. All that we'll be talking about tonight have to do with these oxidation states. One of the oxidation states of oxygen allows it to capture two hydrogens, and this allows it to make molecule of water. This is what it looks like. A water molecule can have also a polar effect. It has a positive side and it has a negative side, which allows it to be rearranged into different structures. And we'll be talking a little bit about that tonight also. But the first thing that probably would be of interest is that in all biology and all life on Earth, we are made out of a network of cells, of interconnected tissues. And these interconnected tissues then also are interconnected with the cells. We have a vast communication network that connects all of our cells in a wonderful and a beautiful, glorious way. This organization and orchestration between cells allows the cells to communicate and to coordinate all of the activities that take place within them. And by activities, I mean molecular activities. So this is where an atomic physicist actually plays. All cells are made out of interconnected molecules, some of them very complex molecules and proteins that carry on the processes of life inside the cell. The one thing that we want to emphasize tonight is that all of this activity takes place in a bath of salt water. And this salt water then interconnects all of the molecules and all of the fields that exist inside living cells. Salt water is the basic fluid of life, the basic vital fluid. that's inside every single cell. And this salt water, made out of water, of course, can be oxidized and reduced. And this area of biology is called redox biochemistry, because the reduction and oxidation of the vital fluids also changes the nature of the chemical reactions that take place inside our cells. So if you were to peer inside a cell where all biochemistry takes place, as I mentioned before, this redox signaling, or reduction and oxidation states, creates a network of signaling in between the molecules that are inside of our cells. And they have special properties and special reasons as in life, there's always a reason for these different reactions. Oxygen, nutrients, and hydration and energy come into our cells through the membrane. And most of the oxygen is introduced into an organelle called the mitochondria. The mitochondria has a very special purpose. It creates the cell. It's sort of like the fireplace, if you want to consider it, of the cell. It creates the energy that the cell produces in the form of ATP, which is a very special molecule that is the fuel for that purpose. Along with that, however, it also creates a series of oxidants, superoxides and hydrogen peroxide and reactive oxygen species, as we had mentioned before, which are different oxidation states of oxygen. And these special oxidants inside the cell have a very important role. As they are produced by the mitochondria, they also are able to float freely around the cell until they encounter one of the antioxidants. So our cells produce oxidants and free radicals, and they then are eliminated by these antioxidants that also are placed in strategic areas in the cell. This has a very special purpose. It creates what they call a homeostatic balance inside the cell. It's a redox balance, redox and oxidation balance, by which the oxidants are able to be reduced and eliminated at the same rate as they are created. It creates an equilibrium or a balance of the oxidants inside the cell. And this is very important, just as a fire, for example, is able to produce smoke. And the smoke and free radicals from the fire can be damaging. If they're able to be controlled inside the cell, then they perform a very important purpose. And that is when the cell is damaged for whatever reason, just as if a fire is damaged or a fire is not ventilated correctly, or there's something in your house that smells like smoke, you immediately know that some damage has taken place. And it's these very oxidants that are produced in excess that create a state called oxidative stress that are able to push the genetic buttons necessary to start the repair and replace mechanisms of the cell. If the cell is not able to repair itself, for example, within about 2 hours, it will be replaced. It'll kill itself and be replaced by healthy, normal cells through cellular division in the neighborhood. I explained just a little bit why this is important. If a damaged cell inside your body is able to survive, then over time it will divide into 2 damaged cells, and 4 damaged cells, 8, 16, etc. And that propagates and spreads the damage throughout the organism. Aging, by the way, is one of the results of such damage that is propagated slowly throughout our system. And so we need to be able to repair these cells. And these smoke signals, if you will, the oxidants and free radicals, are very important parts of the mechanisms that program the cell to repair itself. The cell stops all its activity when it smells, if you want to think of it this way, when it smells smoke or when it has an oxidative stress condition, and starts to undergo the repair and replace process. Many scientists have a specific interest in the redox signaling technology. James Watson, the person who discovered the structure of DNA, inside the body, is now dedicated his entire career to redox biochemistry. And he says there's a delicate balance between two. We're talking about oxidation and reduction reactions. Physical exercise promotes the body to make large amounts of oxidants, molecules called reactive oxygen species that we're talking about. And it helps forge the chemical bonds which stabilize proteins as they fold and also detects damage in the cells. These men, for example, were able to earn a Nobel Prize for their work in the G-protein coupled receptors, which have a great deal to do with the redox signaling. Here's another redox signaling molecule made from nitrogen, from what they call reactive nitrogen species, or nitric oxide is part of those, and they earned the Nobel Prize for that. There's also several books that have been published, journals specifically dedicated to this subject, conferences all over the world. And we're starting to realize for the first time that the antioxidants are not just the things that are found in foods, and they get rid of these terrible oxidants inside of our body. That's actually a myth. What we're starting to understand now is that we need a perfect balance of antioxidants and oxidants in order to be able to create a healthy cell and normal life processes. They work in conjunction. They're partners. They're equal partners. And the most important thing then is that we have a balance between these two things. And they create a signaling network that allow to detect the damaged cells that are inside the body and start the repair process. Without this process, healing would be absolutely impossible. We need a detection, a repair, and replace mechanism. I'm going to go on a little bit, just a little bit deeper, I suppose. This is an interesting experiment that was done with the regeneration of a tadpole tail. There's a tadpole. If you cut their tail off, they'll regenerate their tail. And down at the bottom, the tadpoles are transparent, so you can see through them. They put in fluorescent dyes that were able to detect the oxidation of the tissue, the oxidation states. And if you notice at the bottom, the part of the tail that is regenerating has the highest oxidation. That's in the reds and the yellows in the tail. And then when the tail has finished its regeneration cycle, the oxidants all of a sudden disappear. So the scientists decided, well, we're going to get rid of the oxidants by putting certain antioxidants in the mix and see if that affects regeneration of the tissue of the tail. And they found that when they put certain antioxidants inside a mix, that the tail was not able to regenerate. And so we're finding that this reactive oxygen species actually is important for the regeneration of tissue, for the detection, the repair, and the replacement of tissue. Up to this point, we were only able to do this inside of living animals because mitochondrial ROS, which is required for tissue regeneration, was only able to be produced and stabilized inside living cells. But we have been able to stabilize it outside of living cells and then reintroduce it now into living tissue, and this is quite a discovery. Okay, well, James Watson says that if we can do so, we can use reactive oxygen species to prevent the occurrence of several problems such as type 2 diabetes, dementia, cardiovascular disease. So this is the reason for the ongoing study of this subject. And again, is emphasized that, the reactive oxygen species is an important player, as Yannick says in his paper. It's widely accepted that ROS plays a crucial physiological role in diseases and also in cellular homeostasis, which means balance or in this. So as mentioned before, we are able to take these reactive oxygen species, perfectly balanced set, place them inside a bottle, the same balanced species that exist inside cells. We've done the research that show that these compounds exist and they're completely biocompatible and bioavailable. By the time that I was able to come in contact with this technology, it had already had 16 years of study. It was only able to be produced by a machine. We were able to stabilize it, prove that it's, again, biocompatible and bioactive. And there's a couple of things that we saw right at the beginning. One was that it increased the efficacy of the endogenous, the internal antioxidants inside of our body. Those are not the ones that are taken in by foods, but the ones that our cells produce. It increased the efficacy of those by 400%, and this caught my attention. It also releases NRF2, which starts this defense, repair, and replace mechanism in cells. So it was very interesting. NRF2, by the way, is one of the primary defense mechanisms that the cells have. It releases those as the primary defense mechanism, but also as a repair and replace mechanism. Microbes, by the way, were killed in under 30 seconds with these redox signaling molecules. That was because the body uses these as antimicrobial agents against everything that exists. In fact, the immune system uses these same molecules, these oxidized species, to kill bacteria. But it had no effect on the beneficial flora in our microbiome, in our gut. And we have several human studies that show this. Cell cultures also showed no inflammatory markers, no cytokines, no inflammation, nothing stimulated in living cells. Antioxidant efficiencies increased. It accelerated the kill rate of the cells only in cells that had become damaged past repair. And it increased its sensitivity to some hormones. And in some cases, it actually reversed the aging of cells. The aging of cells on a cellular level, which we call senescence. It has some very important effects. One opened effect is that it opens up the detox channels in our cells. It allows the toxins that are in our blood to make it out through the gut, out through our intestines, and also through our kidneys. It stimulates antioxidants in humans. We know that because the oxidized glutathione that is in our blood goes down. Also, the oxidized LDL cholesterol levels go down in blood in our human trials, which is one of the causes of heart disease. It is the same signaling messenger system that activates stem cell adhesion and differentiation. And it also is a key to start cell regeneration cycles. We have something that's very important. One thing that we found now in 22 years of studies, over 30 studies that have been done both in cells and humans, have shown that there's absolutely no toxicity whatsoever. In one study, for example, in animals, animals were given orally what the equivalent of 4 ounces would be in a human before running on a treadmill, and they found out that animals on a treadmill could run 29%, or these little mice could run 29% longer with ASEA than with just the saltwater placebo that they were given. The human metabolism also undergoes shifts when in this. We checked out 260 indicators of the human metabolism, and over 62 of them shifted in a way that we believe is to preserve energy. So we have mice running longer. We have glycogen depletion, which means our energy reserves are going up. increased antioxidant efficacies, less oxidized glutathione and less oxidized LDL cholesterol in humans, and absolutely, and in animals too, well, in humans also, but in animals also, and zero, about as close to zero effect, toxic effects as you can have with any substance, including salt water itself, saline. We have 50,000 people using it. They have also run studies on skin. And I'm going very quickly here because I'm sure that you have questions. 23% wrinkle depth, reduction in blotches, smoothness, elasticity, etc. of the skin was dramatically improved. Oxygenation of the skin also was increased by over 16%, and the cell turnover, cell regeneration in skin was sped up also, was accelerated again by about 16%. We have also studied the cells that are in the cardiovascular systems extensively, finding that it helps carry messages through our cardiovascular system, the redox signaling messengers. Specifically to find damage in our bodies. It also helps in hormonal balance in almost every aspect of hormonal transmission. We found that it increases the reception of certain hormones, including insulin and the thyroid hormones. The current skin health research also, we already took a look at that. Lung tissue, incredible results on lung tissue. We looked at lung endothelial cells, and in many of these studies, and found a regeneration and increase in glutathione in these cells. Digestive system also, we don't have many studies on this, but we do know that it preserves the natural beneficial microbiome in our digestive system. Nervous system also requires a very careful redox balance. We believe that redox balances help to be maintained, and the reduction of oxidative stress in the brain also causes a reduction of anxiety and certain symptoms. Overall, there is an absolutely incredible results. There are incredible results in many ways, but the thing that impressed me is that this technology has probably the greatest, I believe, will create and fuel the greatest advances in medical science that we've seen so far, because they are directly involved with cellular regeneration, reducing oxidation. They're completely safe. They mediate and regulate many of the mechanisms that restore balance and regenerate tissue, and it optimizes many cell functions. So I am completely absorbed in this technology right now. I think it's the greatest thing in the world. I think I'll turn the time over to Kathleen or to Dan then for questions.
You answered a lot of questions, Dr. Gary, that I didn't think I had. No, that was fantastic. That did clear up a lot for me. And, we try and go out there and share these people with sounding like an atomic medical physicist, obviously, and try and get out there in layman's terms so people understand. But that was incredible for me. We've got Dr. Dayton on. If you guys would like to ask any questions while we've got Dr. Gary Samuelson. Bruce, also, if you'd got any questions, you'd like to unmute yourself, more than welcome. We've got him. This is a very rare situation.
A couple of questions. I've been using the product for not long, maybe a couple of weeks, 2-3 weeks, something like that. And is there one thing that I just personally have attention on? My blood pressure is a little high. And in talking to Kayleen, she said, oftentimes when you start, the blood pressure will go up because the cells are detoxifying, and then it will tend to come back down. Is there any general time frame in which that might occur? And I understand you're not making any commitments that this handles blood pressure or whatever, but I'm assuming it'll help.
No, basically from the medical doctors that have been using this, drinking sufficient water, probably about 10 glasses a day, is good to help this process happen. It will, with the NRF2 activation, throw a lot of toxins into the blood right at the very beginning because it's detoxifying, like you said, your fat tissues and such that have built up. And this can, in a sense, raise blood pressure because the body is trying to get rid of these toxins. And but it, like I said, about 3 weeks or 4 weeks into this, if you're taking about 2 ounces twice a day. How much are you taking?
I'm taking about 8 ounces a day.
8 ounces, so 4 ounces twice a day.
I take 4 ounces in the morning and then two 2-ounce shots later in the day.
Okay, great. Yeah, you should see a normalization in about 3 to 4 weeks. You should start seeing it right now.
Okay, good. That's great to know. And the other question is similar to this, or at least along this line. Is there a process? In other words, do the benefits tend to build up over time?
I've noticed that, yes, they do, because it helps detect and repair damaged cells. Little by little, you start seeing different things happening in your body. Veins, vascular tone, for example, increases in some women visibly. You're able to see your athletic ability and the muscle soreness and such go away if you are athletically inclined. But I noticed that because I used to run track, and when I started taking the product, I was able to run again. And I was able to run long distances and not get sore as I used to before. That for me, that was wonderful.
Yeah, I've noticed. I live on a hill, and so I used to run years ago, but kind of got to my knees and stuff. But I walk up this hill every day. It's a pretty steep hill. I live in the mountains, and so I've noticed that it's easier. Or I'm not getting as much oxygen deficit or whatever that is. It just seems to be improving such that it's easier to get up the hill. Anyway, those were the two main questions I had. I think you've answered them, and if I think of anything else, I'll unmute myself again. Thank you, Doctor.
Oh, great. Thank you, Bruce. Thanks for coming.
Yeah.
One question that has come up, Dr. Samuelson, with some of the folks that we're working with is...
Yeah, a lot of people have issues with heavy metals. What can you tell us about heavy metals? I
think that was Dr. Dayton. Is that right? Yes. What I heard was a little bit muffled, but you said, what about heavy metals, I think, if I heard that right. Yeah, NRF2 and glutathione is a good chelator for heavy metal toxicity. Yes, Dan? And so one of the things that we've seen is that and heavy metals are able to be released from the body when glutathione is activated, when it's more effective. And so we know that ASEA activates glutathione. We know that it activates NRF2 in the body. And so we have also seen an increase in urine heavy metals and increase in other heavy metals when people are taking ASEA, especially for the first time. I'm sorry, did that help answer the question, Dr. Dayton?
I actually asked that, Dr. Gary. No, that was fantastic because we've got a lot of people that do have issues with heavy metals. So, but Dr. Dayton, you're on the line. Would you like to ask any questions while we've got Dr. Gary Samuelson?
Possible. Maybe he can't hear us. I'm not really sure. I have him unmuted, but...
We do have a chat box too if you want to ask any questions in the chat box if you have any problem with your microphone.
One of the things often that comes up, I'll ask a question from health practitioners we are talking to, is if they want to get their own certainty on the science behind it, Dr. Samuelson, because maybe they're not familiar with redox biochemistry, where are good places for them to do their own research?
Okay, well, besides, the general references to reactive oxygen species and redox biochemistry, which are extensive, specific research can be found in the ASEA website and through some of the health professionals. but it's proprietary, and so ASEA releases that as it sees fit. One of the problems that we've had, Kathleen, is that many of the early studies were actually done when this was considered for a pharmaceutical. And so we can't make any claims, and so therefore we can't publish any results from those types of studies. But yes, we can. There have been more than 22 years of studies, and over 30 studies, as I mentioned before, in all sorts of situations. There's one thing that I can vouch for absolutely, that it is absolutely safe. We have not seen not even one indication in any animal or in any person or any subject, one indication of toxicity whatsoever. And so that's one of the things that we can say from the studies. Hopefully that it answers part of your question there, Kathleen.
Definitely. And another thing that comes up too that's confusing when we encourage maybe someone to talk to their doctor about the product in coordination with their other nutrition or body protocol or treatment is it high in sodium, and how do I make sure it's not a diuretic or it's not going to affect my sodium levels?
No. Well, ASEA has about as much sodium as a carrot and a half or 2 carrots. So if you can replace 2 carrots in your diet, you're fine taking ASEA. You know, there's very, very little sodium in ASEA.
Very good.
Dr. Gary, I've heard you talk about before when it comes to addictions. I have a lot of people that do stop smoking in a very short period of time, or reduce their alcohol, or, you know, come off what they're addicted to. And I've heard you talk about something balancing within the body apart from the detox.
Yeah, hallelujah. Congratulations to those people who finally, are trying to turn around. I have a very good friend, Dr. Rob Ward, who is an addiction, ran an addiction clinic for over 15 years. And he said that he was able to have more results in 1 year with ASEA than he did in all 15 years of his practice before, because it helps to reduce the anxiety of these people who are trying to get off of the addictions. So yes, it has a wonderful effect.
And obviously we talk to a lot of health practitioners around the world, are working closely with them just in regards to the results they're getting for patients. And a lot of the naturopaths obviously have got their patients or their clients on a good alkaline diet. And they're saying to us that the results they were getting in 6 months, 12 months, 18 months, they're now getting in 3 weeks, 6 weeks, and 12 weeks.
Yeah. Yeah, I see healing vastly accelerated. Healing that normally takes place in seconds. Normally takes place in minutes, takes place in seconds. So, one thing that I mention once in a while to practitioners is that if you have hemorrhaging or bleeding, ASEA helps to cut down the bleeding or to stop bleeding. And you start to see the closure and processes that normally take minutes that take place in seconds, and then a process that normally take hours to complete take place in just minutes. And so it's fascinating to watch. If you ever have a cut or a lesion of any sort, and you place ASEA on there, you can actually see the results taking place right before your eyes. It's not difficult to see. And that's true, for long-term things too. Some veterans that have scars for 20 years have said, or I've seen, have the scarring actually reduced after several months of taking ASEA. Their scars start to become small white lines, and that's certainly not a placebo effect. That's an actual physiological effect.
We have a question in the chat box from Bruce that's just asking, does the product have to be taken on an empty stomach? Are the results negated if one takes it shortly after eating, or what is the significance on taking it on an empty stomach if that's the most beneficial?
Of course, that's the most beneficial. You get the most absorption. However, ASEA goes through water channels which are absorbed in just minutes anyway, whether or not you have a full stomach. I suggest that you take it 5 minutes before you eat every meal, 5 to 10 minutes before you eat at least. And this just makes sure that your digestive tract and everything is open and ready to absorb the ASEA.
Another question we had at one point is why do the digestive enzymes in the stomach not harm the molecules in any way?
Oh, well, that's a very good question. That was one of my first. So, the question is, why in the world are these molecules so stable? Because they should not be stable as they are. And so we studied those. And they actually have configurations which are combinations of mixture of this proprietary substance that allow it to become stable enough to resist a lot of chemical effects. The enzymes, of course, will break them down, but it won't break them down immediately. It takes several minutes, and in those several minutes, these are absorbed into the body and can have their effect. Now, when we're taking several minutes, well, here's another thing that we've found. In the metabolic, what they call the metabolomics experiments for humans, we specifically looked at the effect on the metabolism over time. And what we found is that after taking 4 ounces only in the morning, the metabolic shifts that we saw were able to be seen up to 24 hours later. So it was being absorbed into the body. It was being, and then it took effect in the metabolism, and there's physiological effects.
That's really interesting. Another question that's come up is when a person is replenishing their molecules with this product, does it ever start to bypass the body's own natural production of redox molecules over time where then the body, is not producing as much or if a person stops taking the product, again, somehow their own natural production of the molecules are diminished?
Yeah, the mitochondrial production of the redox signaling molecules, the natural production, has to do with the metabolism of sugar. So we'd have to slow down the metabolism of sugar. That doesn't happen. In fact, if anything, it shows that maybe the efficiency of the mitochondria to burn fatty acids is increased, not diminished. And so what we'd expect almost is maybe even an increase in the natural production of redox signaling molecules when we ingest the same. So no, there isn't a counter-regulatory effect. There's no dependencies that build up.
We've had people ask if you could actually physically test somebody's natural production of redox signaling molecules and how much they'd be producing at that stage or age in their life.
Yes, you can. Since it's directly proportional to mitochondrial activity, we can see that the mitochondria or the metabolism does slow down over time. In fact, significantly, when you're 10 years old, if you say that's about 100% of mitochondrial activity, by the time you're 70 years old, you're at maybe 20%, if you're lucky, or 30% of the mitochondrial efficacy, which also means that the redox signaling molecules are slower. Cellular regeneration is slower. Autoimmune response is slower. Things kind of slow down as you age, possibly because of the reduction of these molecules in your body.
Sorry, Dr. Dayton's come in with a couple of questions. Has anyone published clinical outcome studies is the first question.
We have no published studies. All are proprietary. ASEA owns the rights to those, and so you'd have to speak directly to the company in order to be able to receive those. There are a lot of things out there.
Is there any direct ration of benefit with the amount taken?
I hear you.
Dr. Dayton asked if is it beneficial, more beneficial taking, say, 8 ounces twice a day than 4 ounces twice a day, actually taking a larger amount?
It depends on really on the topography of the damage that's in your body. If you have large-scale damage in many areas of your body, it sometimes takes much more. So we've seen that up to 8 ounces twice a day is effective. If you have large-scale damage, it seems to be more effective. For normal, healthy people, usually only about 2 ounces twice a day is fine. For some people who are trying to detoxify, at times it's a good idea to sometimes go below that amount and take smaller amounts more often during the day. So it really depends on the person. But the general rule is if you can't tolerate it very well, take less. If it seems to not have exactly the effect that you need, take more, at least for a few days, up to maybe 8 ounces twice a day, and see if your body reacts. One thing that you'll find if your body starts to react, that the immune system starts to attack the damaged cells, which takes a lot of energy. So you might find lack of energy. You also might find that there's a detox effect. And you drink a lot of water, and in almost all cases that takes care of those problems.
Dr. Samuelson, is there I've had questions where, is there a saturation point? Is there too much that you can take where your body then just becomes saturated with these molecules? Yeah.
I mean, some people take two bottles a day or something like that. But becoming saturated is something that I don't think is possible, because your body just has enzymes that break this down. So the excess is broken back down into salt and water. And the only effect that you have is a drain on your wallet.
Well said. And, sometimes when a person starts replenishing their molecules, if they do have a health challenge or a condition, they'll visibly see it get worse or physically feel it get worse. How would you help them understand what their body's going through or prepare for that to potentially happen?
If you have ever come back from a period of sickness, and you realize that the kids have destroyed your house, you'll realize that, when you finally have energy and the vision to clean up, it'll take a little bit of energy. But not only that, you'll have accumulation of garbage, and you'll have to throw that garbage out. The body is very much like that. When you've been sick for a while or inactive for a while, and the body realizes is trying to clean up, you will feel a little bit drained, and then of course the detox as it throws as your cells start to throw the garbage out. Drinking a lot of water will be helpful in that case, but it does get a little bit worse before it gets better, and that's just the nature of things.
Well, we're coming up on the hour. John Spagnola, I know you're on the line. I don't know if you've got any questions before we wrap up and send this incredible man back to his beautiful wife. I think,
John,
I think we might have Dr. Dayton. Bruce, any more questions before we let Dr. Gary Samuelson go?
I was just going to say I've unmuted John Spagnola. So, John, you should be able to talk.
Oh, thanks. I didn't know I was unmuted. Dr. Samuelson, can you hear me?
Hello, John. Yes, I can hear you.
Yes, thank you very much. I found the presentation very informative. It was stimulating, and it answered a lot of questions that I did have. I've been having quite a bit of success with addressing scars, a sebaceous cyst, gone as a result of the gel, and been pleased with that. So can you address the situation of it affecting maybe drug withdrawals? That's possible?
Withdrawal symptoms, is that right?
Yes, I'd be interested in that.
Yes. Well, one of the problems with toxicity in the brain and neural inflammation is that there's oxidative stress buildup in the emotional centers of the brain, which lock the body into the stress response. reduction of oxidative stress is something that ASEA is very good with. The redox signaling molecules are very good with. So in reducing the oxidative stress in the emotional centers, it also helps to reduce the anxiety and the stress response, helps the body to get back into normal hormonal balance also. And we've found, as I mentioned before, through doctors that have been in drug rehabilitation, specifically, rehabilitation from prescription drugs and such, that it takes the edge off, so that they can sleep during the nights and they can feel at ease, and that helps. Of course, you also need a great network of caring people that will help the people through their addiction cycles. That's important also, as you know.
Yeah, that's appreciated. Does exercise, would exercise help with, absorption or the circulation of the molecules, or somehow augmenting the effects?
Exercise is the only thing that can produce these molecules inside your body. So, this is what James Watson has been doing. He's been spending billions of dollars, he's trying to raise billions of dollars to see how the effects of different types of exercise can increase the amount of reactive oxygen species and these molecules inside your body. He isn't aware that we have stabilized these and that they can be used. I don't know if he ever will become aware. I'm not sure. But our clinical studies with humans show that when they take this, the metabolic balances are that in the people that have taken ASEA are similar to those that have exercised for several minutes, if not hours. And so the answer is yes. Exercise does increase these molecules. It increases cell regeneration, and it's wonderful.
Thank you. Last question that I can think of right now is, how about diet? Are there certain diets that would augment the effects of the redox molecules? Sugar, for instance, or high carbohydrates tend to have a limiting or problematic effect at all?
Yeah, they have a detrimental effect on this. Actually, if you're going to take the redox signaling molecules and you want the maximal effect, of course, you need to also have a good diet, which I say is basically lots of greens, lots of vegetables, lots of multicolored vegetables. In fact, as many colors as you can get on your plate. Learn how to cook them. A little bit of meat is okay. And that basically, I think, is the best diet. High carb diets and such create an excess of the molecules. In fact, so much of an excess that they cause damage. So they flip the balance the other way, where you have chronic oxidative stress in your veins and in your pancreas and your organs and such. Due to too high of a level of glycemic index in your blood.
That was very helpful. I appreciate that. I'm turning it back over to Kayleen.
Thanks, John. I also know that we have another caller that I want to apologize. You've been muted with the area code 734. So you're unmuted now if you'd like to ask a question.
Oh, you can hear me?
Yes, we can hear you now.
Oh, okay. This is Nick. Okay, I heard about the intestinal tract. This is very exciting because I don't know how many millions of people have intestinal problems. And you mentioned about the microorganisms, the beneficial microorganisms staying alive in the intestinal tract. Have you done any testing on that? And the reason being, my cousin is a paraplegic, a T12, so he basically, from the belly button down, he has no feeling. And for years, every time he goes to the bathroom, he would lose blood. And that's a combination of hemorrhoids and a lot of spitting. Well, after 3 days, there hasn't been a drop, not a drop of blood. And that started, we started him on that, I believe, 3 weeks ago. And so what's even more exciting is he hasn't been able to control his bowel movement. So he has accidents, a lot of accidents. And this has been a problem because he just crossed France a few years ago and climbed the Eiffel Tower, and he always, he has accidents. For almost every single day for the last week, he hasn't had an accident. So I'm blown away. Like, you definitely have my attention. And just wondering, has there been any studies done on the bacteria? Because there's so much going on in there with the hairs inside of the intestine. Are we rebuilding anything, or are we just repairing skin tissue? Any information? Go ahead.
Okay, great. No, with ASEA itself, we have a large clinical study which looked at the microbiome before and after taking 4 ounces a day of ASEA, and found that there was absolutely no difference in microbiome between people that were taking ASEA and people that were not, or before and after either case. So it tends to show that there's no negative effects. However, it is antimicrobial, and we ask, well, how does the body tell a difference? Somehow the body knows the difference between, the beneficial flora and the ones that are not, and tends to protect the beneficial flora. I've been working with a scientist the last couple of years now that has focused on the health of the gut. And I wish I could tell you some of the things that are going on, but there are tight binding proteins inside the gut that hold these cells together. Gluten tends to tear these tight binding proteins apart, as well as does glyphosate and some insecticides and herbicides. They tear these things apart, so there's large damage in the intestines. But there's also leaks. And the leaky gut allows toxins and larger molecules to get into the blood and causes inflammatory reactions in the gut and et cetera, which can be manifest by bleeding and such. When these tissues are able to then regenerate faster than they degenerate, we're winning the battle. So it's able to help regenerate tissue faster than it's degenerating. There are also carbon-based molecules that can help restore these tight binding molecules in the gut. There's a lot of things that we can do to work on the gut these days.
That's very exciting. One more. In plant science, I've worked with some of the national crop growing champions that grow the most corn and the most soybeans here in the United States. And we took a microscope and we're looking at the beneficial bacteria in the soil profile. And so we put it against plant roots. And what we found was the plant would excrete water through the roots. The beneficial bacteria would absorb it, go through their life cycle and produce a few hundred babies and then die, and then the plant would absorb the dead bacteria. And what we found was yield, the health of a crop, the yield is determined by the beneficial bacteria in the root system. So it's basically just like the human intestine, and I'm just wondering, and I can't find any research on this, but if we strengthen the beneficial bacteria and we actually ingest the bacteria into our bloodstream, and that's a food source for rebuilding the body, I'm just wondering if there's any connection there. And has there been any work like that done? We have a lot of theories. We have a lot of theories on that. I'm just wondering.
Yes, no, you are absolutely right. The ecology of the bacteria in our body is absolutely essential. We only have 30,000 genes at most in our body, which means we can only produce about 30,000 proteins. But yet we use and utilize over 200,000 proteins in our body. Where does the other 170,000 come from? And the answer is the bacteria. The bacteria genome actually is more important to produce proteins in our body than the genomic makeup of our own body itself. We're more bacterial proteins than we are human. So it's very interesting. Yes, the microbiome that we have inside of ourselves is absolutely essential to keep that balance. And as we destroy the bacteria through our agricultural processes, we're also destroying the nutrients that we have available in our foods. And it's a very serious concern, and I'm glad that you brought it up tonight.
Well, that's exciting. You just made my night. I'll tell you why, because I've had this theory for a number of years. Because we had a large livestock operation. What happened to the animals, they had a disease, and they could no longer absorb enough nutrients. They were protein deficient, even though they had plenty in their diet. And what we discovered through postmortems was that those hairs in the intestine were destroyed.
Yes.
And so now if we can rebuild those, I don't know how many people on the planet have diarrhea problems because their intestinal walls have been destroyed. And then on top of that, you think about our diet, the number of preservatives like MSG that are in our foods that actually kill or mutate the beneficial bacteria in our intestines. So if you take ASEA and we start reversing this, okay, you made my night.
Well, could you do me
40,000 extra protein. Yeah, go ahead.
Could you do me a favor? If Dan or Kathleen could take your email, I can send you then quite a bit of information on and studies on this.
Good. That'd be great.
Yeah, Nick, I can get we can talk and I'll get your email and send that to Dr. Samuelson, okay?
Okay, I appreciate it. Thank you.
Yeah.
One more question from Dr. Dayton, Dr. Samuelson, just one more question. With topping up with these molecules, what are the effects on the issue where the immune system's attacking the body? And the other issue, which is a situation I was in, which was the worst case scenario for anybody on the planet.
Wonderfully enough, the immune system has a detection mechanism which is able to distinguish between damaged cells and healthy normal cells. When that detection mechanism breaks down, then it will actually get its signals crossed and start attacking healthy cells. And that's an autoimmune problem. So what we have seen with ASEA is that it helps the communication system. I call it the cell phone, to become more clear. So you get more bars on your cell phones, the communication is clear, and there's a better distinction between damaged and healthy cells. The body, when taken with ASEA, will actually be biased against the damaged cells, attack the damaged cells as it should, and then help to protect and regenerate the healthy cells. And that's a natural process. ASEA helps that process along. Does that do it for you, Dan?
Yeah, I know that when anybody asks a question, when it comes to what about this issue or that issue, we ask the question that you ask back, is there a cell involved?
Yeah. Well, it's remarkable. This is the very process that helps the cells to recognize and repair the damage that takes place. Here's the way I think of it. If we were able to repair our damaged cells, what does that mean? What does that mean to our health?
That's well said.
Yeah, absolutely.
Oh, good. Yeah, are you still with us? I wanted to thank you, Dr. Samuelson.
Would you believe he's dropped off? He's done all that hard work and he's dropped off.
We were going to acknowledge him and say, thank you for your dedication to this incredible science, because for those of you just meeting him for the first time, he's dedicated a significant part of his life to helping us really understand what these molecules are and making sure they were stabilized so that we could use them as a resource. So definitely continue to tune in and learn as much as you can about redox biochemistry and ASEA itself, and ask questions of the folks that are sharing more information about it with you as well. Anything else you'd like to say to the crew tonight?
Yeah, thanks for coming on, everybody. Dr. Gary Samuelson, I mean, he's dedicated his life to helping humanity. Because of him, I'm here. which is fantastic. But there's a lot of people out there that shouldn't be here that are still, and he's done an incredible job of not only with redox CV molecules and what he's done there, but he continues his research to help people in however way he can. So thanks for coming on, guys. Really appreciate it.
And with that note, we'll say goodbye to everyone. Have a good night.